So good evening everyone, and sorry for a delay. I just forget about the timetable, right? So can we start now? It's all right? Audible, visible? Good evening, good evening, good evening. So please help me up to where we discussed in the last class. Very good evening, very good evening. Tell me up to where we discussed in the last class. Please pin your answer. Cell introduction done. Up to where it done? Cell introduction done up to where? Cell theory started or not? Cell theory started or not started? Not started. Cell definition completed. Yeah. So we are starting with cell theory. Have a look now. Cell theory. So if we talk about cell theory, you keep one thing in mind. It is given by Sleden and Swan. Sleden and Swan. So if you talk about Sleden, he was a German botanist. And if you talk about Swan, he was a British zoologist. Is it clear? Right. So, what observed by the Sleden? Sleden observed so many different plants. He observed algae. He observed fungi. Now fungi is not a plant. Bryophyta, pteridophyta, gymnosperm, angiosperm, monocot, dicot, dwarf plant, tall plant, flowering plant, non-flowering plant. So many plants he observed. And he find one common thing in every plant. And what common thing he observed in every plant, what is there? So he just write his finding and conclusion after observing the different plant, and Sleden told all plants are made up of cell. So plants are so many of different kind. They may appear different in their structure. Variations is there in the structure. What is common in every plant? Every plant, they told all plants are made up of cell which form tissue in plant. These are the finding of which one? Sleden. Is it done? So Sleden told all plants are made up of cell. Beside of that, plants are different from each other in their structure, but one common thing is there. They are made up of cell, and these cells constitute which one? Tissue of the plant. So you got the idea about the finding of the Sleden. Now I am going to tell about what told by the British zoologist, and that is which one? Swan. So what told by the Swan? Swan observed so many different animals, and based on his observation, what he concluded, he told in animal outermost boundary of cell is thin and membranous. Thin and membranous boundary is present in which one? Animal. Got the idea? So that observation given by Swan after studying so many different animal cell. He told whatever the animal cell doesn't matter. But if it is an animal cell, if it is present in animal, their outermost boundary is which one? Their outermost boundary is which one? Thin. Their outermost boundary is which one? Thin and membranous. Nowadays, for this we called, nowadays it is called plasma membrane. Nowadays called plasma membrane. But this plasma membrane word not used by whom? This plasma membrane word not used by Swan. He told outermost boundary is thin and membranous. Clarity came in the mind or not? Is it done? So this is the finding of which one? British zoologist. Now we are moving ahead. Swan also told a very important line. What told by the Swan? He told presence of cell wall is an unique character of plant. Who is unique in plant? Cell wall is unique in plant. So Swan not only study what? Animal cell. He also study which one? Plant cell. He told in animal cell, outermost boundary is thin, and in plant cell, who is present? Cell wall is present. So in plant cell, cell wall is present. That is told by whom? Swan. Later on, what happened? Let me tell you. Swan give the hypothesis of cell theory. Gives the hypothesis of cell theory as what he told? All plants and animal are made up of cells and their product. So examiner can ask, who told all plants and animals are made up of cell and their product? Then what the answer will you give? Who told all plants and animals are made up of cell and their product? Who give this hypothesis? The answer is only Swan. But if they are asking, cell theory is given by? Cell theory is given by? You have to write which one? Sleeden and Swan. Both. Got the idea? But these two gentlemen was not aware about how new cell are arising. How new cell arise. Who don't have idea about it? These Sleeden and Swan, they don't have any idea about how new cell arise. So the answer of this question is given by which one? Whom? Rudolf Virchow. So what told by Rudolf Virchow? Rudolf Virchow told omnis cellula e cellula. Its English translation is, of course it is not in English, English translation is all cell arise from pre-existing cell. All cell arise from pre-existing cell, omnicellula, e-cellula, told by whom? Rudolf Virchow. Later on, he modified the cell theory. Who modified the cell theory? Rudolf Virchow modified the cell theory. As what he told first thing in the cell theory, all organisms are made up of cell and its product. Means these are the line he have taken from whose finding? Sleeden and Swan finding, and second line he had their own finding, that is what? All cell arise from pre-existing cell. So now these two lines, both the findings are now included in the cell theory. So the present day cell theory is this. So in present day cell theory is like this. Present day cell theory. So if someone asks you who modified the cell theory, Rudolf Virchow. If examiner asks who added omnicellula, e-cellula in the cell theory, Rudolf Virchow. Getting the idea? Present day cell theory is given by Rudolf Virchow. Cell theory given by Sleeden and Swan. Are you getting the idea? Is it clear in the mind? Is it all right? Done. So who is over? Cell theory is over now. Understanding the idea? Clarity came. Now we are going to talk about an overview of the cell. The next paragraph of the NCERT. So whenever you read NCERT, in NCERT it is written like, when you go for this paragraph of the NCERT, they wrote like three things. I am giving you a glimpse of it. They are writing about prokaryotic and eukaryotic cell. They are writing something about difference between plant cell and animal cell. They have written about shape and size of the cell. So we are going to deal it separately. So first of all, we are going to talk about which one? Prokaryotic cell and eukaryotic cell comparative account. Eukaryotic cell comparative account. This is written in overview of the cell. When you will read, you will find these differences. So I am simply somehow amplifying it. Give your attention. So prokaryotic cell, have a look here. Pro means before and karyon means nucleus. So all the cell who develop before evolution of nucleus are called prokaryotic cell. So who is absent in prokaryotic cell? Nucleus is absent. Who is absent? Nucleus is absent in prokaryotic cell. Eu means true. Karyon means nucleus. So true nucleus means nucleus is present in eukaryotic cell. Got the idea? So in prokaryotic cell, nucleus is absent. In eukaryotic cell, nucleus is present. This is the biggest difference between prokaryotic cell and eukaryotic cell. Now you will ask a question. If nucleus is absent, then what is present? Genetic material is not there. So yes, it is there. So in prokaryotic cell, genetic material. And who is the genetic material? That is DNA is single means one, circular, necked and double stranded structure. Double stranded, and these structure present in cytoplasm. But in case of eukaryotic cell, genetic material is organized into chromosome. And present inside of nucleus. Got the idea? I am just telling you through a diagram, you will understand it better. So what I wrote in prokaryotic cell, genetic material. What is the genetic material? What is the name of genetic material? DNA. This DNA is single. Single means how many? One. Circular, naked, double stranded and present in cytoplasm. Let me draw a diagram for it, you will understand it better. Give your attention. This is a prokaryotic cell. And this is the DNA, genetic material of the prokaryotic cell. How many DNA is there? One DNA is there. Linear DNA is there or circular DNA is there? Tell me, linear is there or circular is there? Circular DNA is there, double stranded there, naked is this. Getting the things or not? And this structure is called, this genetic material here in prokaryotic is called nucleoid. Another name is genophore, or it is also called incipient nucleus. Or it is called which nucleus? Incipient nucleus. Nucleoid or genophore or which one? Incipient nucleus. Understanding the idea, picture came or not came? Clarity came? Is it? But if you talk about the eukaryotic cell, I am drawing a eukaryotic cell, try to understand. This is a eukaryotic cell. I am drawing a nucleus as well. It is a double membrane bounded structure. So I am drawing the nucleus as well. Getting the idea? And who is there? Who is there? DNA, genetic material is organized into chromosome. It is organized into chromosome. It is chromatin. It is chromatin present in interphasic nucleus, na? And when these chromatin condense, it is called chromosome. It is called chromosome. I will tell you later how they are going to occur. Right now I am telling you the basic with difference between prokaryote and eukaryote. Now in prokaryotic cell, who is absent? Histone protein absent. And in eukaryotic cell, who is present? Histone protein present. And due to absence of histone protein, we are telling prokaryotic genetic material as what? Naked. Getting the things or not? So this is genophore. These are called what basically? Main DNA or genome of prokaryotic cell. So the main genome of the prokaryotic cell, main DNA of the prokaryotic cell is how many? Single. So if someone asks you a question, in bacteria how many DNA is there? If someone asks you a question, in bacteria how many DNA is present? One, two, many, variable from bacteria to bacteria. What the answer of this question you will give? What the answer of this question you will give? One DNA. In bacteria, how many DNA is there? One DNA is there. And I am asking some common sense based question. Bacteria is haploid or diploid? N or 2N? So answer will be what? N. Because pair of DNA is not there. Pair of chromosome is not there. So in bacteria, how many DNA is there? One DNA or even one chromosome is there. But that chromosome is not a true chromosome. For DNA, we are saying the word chromosome. So if someone asking you DNA or chromosome in a bacteria is how many? So you answer what? Picture came? Understanding came? Can I say some more difference between prokaryotic and eukaryotic cell? So it is called incipient nucleus, nucleoid or genophore that is present in prokaryotic cell, and this is a well-defined nucleus present in which cell? Eukaryotic cell. So about nucleus of the prokaryotic and eukaryotic cell it is over. Now we are going to talk some more differences between prokaryotic and eukaryotic cell. Give your attention. So if you talk about the prokaryotic cell, in prokaryotic cell, membrane bounded cell organelle are absent. In prokaryotic cell, who is not present? Membrane bounded cell organelle are absent. That is mitochondria, plastid, ER, endoplasmic reticulum, Golgi body, vacuole, lysosome, etc. These all are what basically? Absent in prokaryotic cell, but all these structure are present in which cell? Eukaryotic cell. So you got another difference between prokaryotic cell and eukaryotic cell or not? The membrane bounded structure is absent in prokaryotic cell. If you look at the prokaryotic cell, compartmentalization absent. Compartmentalization absent in prokaryotic cell, and these compartmentalization present in eukaryotic cell. Do you know the meaning of compartmentalization? You look the prokaryotic cell. I am drawing a diagram of a prokaryotic cell. Have a look here. So in this prokaryotic cell, how many chamber is there? How many compartment is there? It is only one compartment. That is the cell, entire cell. Here it is not a compartment, it is a DNA. It is not any membrane are there. But if you look at eukaryotic cell, here who is there? One compartment is nucleus, here is membrane. Another compartment is endoplasmic reticulum. Are you getting the idea or not? Another compartment is which one? Golgi apparatus. Many compartments are there in the cell. Every membrane bounded structure is a compartment. Another compartment is which one? Mitochondria. Understanding the idea? Another compartment is, you can say, about what basically? Plastid. Able to understand or not? So these are so many compartments present in which one? Eukaryotic cell. Many, all organelle making a compartment. So here, and who have a membrane especially, all organelle who have membrane as making a compartment. So these all are compartment of eukaryotic cell. But these prokaryotic cell, this compartment is absent. You don't think it is a compartment, it is the DNA. Understanding the idea, na? So no membranous structure is present, na? Inside the plasma membrane, no membranous structure is there. So that is the reason I wrote compartmentalization is absent in prokaryote, and these compartmentalization is present in Eukaryote. I hope you understand the meaning of the compartmentalization. Now we are going to talk some more difference between prokaryotic and eukaryotic cell, and that is what? Here ribosome present. So ribosome is 70S type. And in eukaryote, which kind of ribosome is there? Both 70 and 80S type. Why I am writing like this? 70S in which one? Mitochondria. And 70S in which one? Chloroplast. And 80S in which one? Cytoplasm. Understanding the idea? So here both 70 and 80S ribosomes are there. Here only which one is there? When you deal superficially like a small children, you will read 70S type in which one? Prokaryotic cell. 80S type in which one? Eukaryotic cell. Understanding the idea? Is it clear in the mind? Now we are going to talk about some more difference between prokaryotic cell and eukaryotic cell. If you talk about prokaryotic cell, cytoskeleton absent. Cytoskeleton absent in prokaryotic cell. In eukaryotic cell, cytoskeleton present. Cytoskeleton present. Here in prokaryotic cell, this cytoskeleton is absent. So you got some basic differences between prokaryotic cell and eukaryotic cell or not. So these are some of the basic difference between prokaryotic cell and eukaryotic cell. Is it clear in the mind? Now we are going to move ahead. Now I am asking a question to all of you, from all of you people. Just try to answer it. The question is, cytoskeleton means, one of the student writing, cytoskeleton is microtubule, microfilament, intermediate filament. These all provide the shape to the cell. These are called cytoskeleton. So they are present in which one? Eukaryotic cell. These all structure absent in which one? Prokaryotic cell. And these have a main role in, we will talk about detail about cytoskeleton when I will teach it, the respective topic. Understanding the idea? Is it clear in the mind? Right. Now I am asking a question. Tell me some common things which is present in both prokaryotic and eukaryotic cell. Commonly present in both prokaryotic and eukaryotic cell. What is commonly present in both prokaryotic and eukaryotic cell? One structure is ribosome. Another structure is plasma membrane. Another structure is cytoplasm. Another structure is DNA as genetic material. DNA as genetic material. It is commonly present in which one? Both prokaryotic and eukaryotic cell. Are you getting the idea? Clarity came in the mind. So who are a common structure in both prokaryotic and eukaryotic cell? Ribosome, plasma membrane, cytoplasm, and DNA as genetic material. And you don't forget, either it is a prokaryotic or eukaryotic cell, cytoplasm is the main arena of cellular activity. So, who is the main arena of cellular activity? Examiner can ask this line also. Who is the main arena of the cellular activity? Cytoplasm is the main arena of cellular activity. Is it clear in the mind? Understanding came? Right. Now we are going to talk about, so prokaryote and eukaryote over. Now in the same, an overview of the cell, plant have, NCERT have written some difference between plant cell and animal cell. And I know you are studying from 9th onward. So I am, beside of that, I am going to tell. What is written in NCERT we not left. So we are going to talk about which one? One side which cell? Plant cell. Not big thing going to ask in NEET. But you don't afford to leave anything which is written in NCERT. So one side I am writing plant cell, another side I am writing animal cell. Right. So tell me the difference between plant cell and animal cell. So you will say, sir, plant cell wall present. One difference is there or not? And here in animal cell, who is absent? Cell wall absent. Cell wall absent. You can say in plant cell G, plastid present. Plastid present. In animal cell, what happened? It is absent. You can say in plant cell, centrosome and centriole usually absent. But this centrosome and centriole is present in which one? Animal cell. In plant cell, large central vacuole present. Central vacuole present. Here in animal cell, large central vacuole absent. In plant cell, less number of mitochondria present. In animal cell, more number of mitochondria present. More number of mitochondria present. If you talk about plant cell, nucleus is peripheral in position. And in animal cell, nucleus is usually centrally located. Agree or not? And if you want to draw a structure, you can say like this. This is a plant cell, assume, but inside the plant cell, major area occupied by large central vacuole. So who is in periphery? Cytoplasm is in periphery. Even nucleus present in peripheral position. Getting the idea? But if you draw an animal cell, what you are finding, कि most of the animal cells, suppose that this is the cell membrane, who is centrally located? Nucleus is centrally located, and between cell membrane and nucleus, who is there? Cytoplasm is there. Understanding the idea or not? So these are the some of the basic difference between whom? Plant cell and animal cell. You got the basic idea? You got the basic idea? Is it clear in the mind? So what differences we have seen? See once again. Plant cell, cell wall present, animal cell, cell wall absent. See, NEET is a game of revision. As many times you will revise, your perfection will increase, your reflexes will develop. Nothing rocket science is going to ask. Plastid present, plastid absent. Centrosome and centriole usually absent in plant cell, here it is present. Large central vacuole present in plant cell, absent in animal cell. Less number of mitochondria present in plant cell, more number of mitochondria present in animal cell. Nucleus is peripheral in plant cell usually, and here it is what? Centrally located nucleus is there. So this is the difference between which one? Plant cell and animal cell. Now we are going to deal a different heading, and that is which one? Shape and size of the cell. Shape and size of cell. Got the idea? So first I am starting, cell have different variety of shape who have different variety of shape? Cell have different variety of shape to perform different function. Means I want to say that is function of a cell depend on its shape. It may be columnar, may be cuboidal, may be irregular, may be spherical, may be oval, may be polygonal. So many different cell structure are there. But a particular structure is adapted for a particular type of functions. Like in animal tissue you study na cuboidal epithelium, columnar epithelium. Both have a different nature of function because both the cells are what? Different to each other. Understanding the idea or not? So what I am saying, the function of the cell depend upon its shape. Getting the things or not? Now I am going to talk about size of the cell. And I just want to take going for a test. Close your NCERT. I am writing the name of some of the organism. Without looking at NCERT, you have to tell me the data, shape, size data. So I am writing it with a star mark virus. Why I am putting a star mark? Because it is not a cell. Virus, PPLO, mycoplasma, bacterial cell, typical eukaryotic cell, nerve cell and ostrich egg. So say about data written in NCERT. I don't want any additional data. NCERT, what data is given about the size of the virus? Virus is not a cell, but beside of that we are telling it is a non-cellular structure. So what is the size of the virus? Without NCERT, 0.2 micrometer. PPLO, right? 0.1 micrometer. Mycoplasma, 0. I'm just checking how many of you are writing. Someone writing, someone not writing. 0.3 micrometer. Bacterial cell, what is the value of bacterial cell size? And one cell I want to write more here, mammalian RBC. Mammalian RBC. Tell me the cell of size of the bacterial cell. Bacterial cell, fast, write bacterial cell. Bacterial cell, 3 to 5 micrometer. And mammalian RBC? Mammalian RBC, here it is bacterial cell 0.3 to 0.5, not 3 to 5. And mammalian RBC is 0.7 micrometer. Typical eukaryotic cell. Typical eukaryotic cell. Typical eukaryotic cell. Bacterial cell 0.3 to 0.5 or bacterial cell 3 to 5 micrometer. Pin your answer. It is 3 to 5 micrometer. And typical eukaryotic cell 0.7 or 7 micrometer. Mammalian RBC 0.7 micrometer or 7 micrometer? It is 7 micrometer. And tell me about typical eukaryotic cell. It is how much micrometer? 10 to 20 micrometer. Who is the longest cell? Nerve cell is the longest cell. And who is the largest cell? Ostrich egg is the largest cell. So it is in ascending order of their size. Examiner will ask. It is in ascending order of your size. Virus is the smallest, 0.0222 micrometer. Magapit, PPLO, 0.1 micrometer. Mycoplasma, 0.3 micrometer. Bacterial cell, 3 to 5 micrometer. Mammalian RBC, 7 micrometer. Typical eukaryotic cell, 10 to 20 micrometer. Longest cell, nerve cell. Largest cell, ostrich egg. Now I am asking a question. Out of these number of cells, how many are visible by your naked eye? How many are visible by your naked eye? One student writing no one. No, this is visible by your naked eye. Ostrich egg. Have you seen the ostrich egg? Have you seen the ostrich egg? It is big size like this. You can easily visualize it. If you can see the chicken egg, of course, ostrich egg is too bigger. It is like this bigger one. Understand the idea? Now, what is the minimum size your naked eye can visualize without microscope? Anybody can pin the answer? See, in my skin, your skin, thousands of bacteria are there, but I am able to see it? No. It is visualized to me? No. I am not finding any bacteria is there. Not a single bacteria I am finding. It is there. Beside of that, our eye not able to catch it. Why our eye is not able to catch it? What is the size of bacteria? What is the size of a typical bacteria? How much? 3 to 5 micrometer. And our eye cannot see anything smaller than 100 micrometer. So our eye either can see greater than or equal to 100 micrometer. Our naked eye can see this size. Less than this size is not visible by our eye. So even a typical eukaryotic cell, 10 to 20 micrometer is there. Means if I remove a single cell from my body, you cannot see with your naked eye. Your blood cell is there, na RBC. When your blood came from your body, you can removing 1 RBC, 2 RBC, 3 RBC, 4 RBC. It is visible to you? Not visible to you. Why? Because RBC size is what? A typical mammalian RBC size is what basically? 7 micrometer. 7 micrometer. Understanding the idea or not? And your eye cannot see anything less than 100 micro. It is more than 10 times smaller. Understanding the idea? Clarity came or not? And now why I am talking much about shape? Examiner can ask a question. Statement number 1. A bacterial cell is larger than RBC. True or false? True or false? It may be given in the paper. A bacterial cell is larger than RBC. True or false? Answer the question. So size of the bacterial cell is what? 3 to 5 micrometer. RBC size is which one? 7 micrometer. Who is bigger? RBC is bigger. So there is a scope of question or not? A scope of question is there, na? Every line have a scope of question. Understanding the idea? And they can arrange the following according to decreasing order of their size. Decreasing order of their size. Now, $1 million question student ask. I am telling you what $1 million question student asked. They came with a doubt. Sir, look, have you seen the NCRT? In NCRT, sir, mycoplasma writing 0.3 micrometer. And PPLO writing 0.1 micrometer. PPLO is a mycoplasma, na sir. Then if it came in exam, what to do? I think probably you also have the same doubt. Kisi PPLO is mycoplasma. NCRT wrote for mycoplasma 0.3 micrometer. And for PPLO NCRT writing which one? 0.1 micrometer. PPLO is what? Mycoplasma. You have also the same question. You have a doubt. Should I give a clarity or it is all right? You want a clarity? Why it is written like this? Ki for mycoplasma written 0.3 and for PPLO it is written which one? 0.1. Why they wrote like this? Let me tell you. I am asking a question. Clarity will come. I am asking what is the size of mammalia? Answer it. What is the size of mammal? You can say, what nonsense question, sir. Mammal, rat is also a mammal, elephant is also a mammal. Whose size you want to know? You are asking mammals, thousands of animals are there in the mammal. You are asking rat size or you are asking elephant size? There is a lot of huge differences in the size. So like mammalia, mycoplasma is also a group of plant. Like it is a class. Mycoplasma is a class of bacteria. And that class of bacteria have many bacteria. PPLO is an example of mycoplasma. So like rat is an example of mammalia, PPLO is an example of mycoplasma. So when they wrote mycoplasma size, they wrote the average size of the mycoplasma. Many are there, na? So they wrote the mean, average. So they wrote the mycoplasma average size. Like mammalia average size. Take the smallest mammal, take the longest mammal, largest mammal, make a mean of it. Average size of mammal we can write. But if you say size of rat is different from the size of average size of the mammal, na? So they wrote the mycoplasma, a class name. Mycoplasma is not a name of an animal. Mycoplasma is a name of a class. Mammalia is the name of anyone? It is a name of group, na? Human came in that, tiger also came in that, lion also came in that, goat came in that, buffalo came in that, blue whale also came in that. Understanding the idea or not? Clarity came in the mind. So PPLO is an example of which one? Mycoplasma. And it is the smallest known organism. Is it clear? So if they are writing PPLO, you write which data? 0.1. If they are asking mycoplasma, write which one? 0.3. Understanding came, picture came, size over, clear, done. Because a lot of debate, a lot of question people arise. Sir, PPLO, mycoplasma, what to do if it came in exam? Now done. So say when overview of the cell is done, cell theory is done, size and shape is done, prokaryotic and eukaryotic cell differences done, plant cell, animal cell differences done. Now we are starting with prokaryotic cell. Okay, ji, have a look. Prokaryotic cell in detail. So, where prokaryotic cells are present? All the members of kingdom Monera have prokaryotic cell. Have which particular cell? Prokaryotic cell. Is it clear? All the member of kingdom Monera have prokaryotic cell. So some of the example where prokaryotic cell is present. So kingdom Monera have only one example, and that is what? Bacteria. Kingdom Monera have only one example. You recall your biological classification. There it is written, na? Bacteria is the sole representative of kingdom Monera. That line you are able to recall or forget? Tell me. Bacteria is the sole representative of which one? Kingdom Monera. Who included in Kingdom Monera? Bacteria included in Kingdom Monera. So what is the example of prokaryotic cell? Bacteria is the example of prokaryotic cell. If you want to write more number of example, you can write many type of bacteria. So what I am writing? I am writing on board many type of bacteria. Many type of bacteria. So Archaebacteria, Eubacteria, these all are a type of bacteria. Cyanobacteria, Mycoplasma. Mycoplasma is also a kind of bacteria. But who is absent in Mycoplasma? Cell wall not present. Cell wall not present in Mycoplasma. Another type of group of bacteria is what? Another group of bacteria is what? Actinomycetes. Don't confuse with Ascomycetes. Actinomycetes is also called filamentous bacteria. List is much more longer, but I am stopping it here. List is much more longer, so I am writing some of the category of bacteria. Archaebacteria, Eubacteria, Cyanobacteria, also called blue-green algae. Mycoplasma, cell wall not present. This Mycoplasma is called joker of plant kingdom. Actinomycetes, it is filamentous bacteria. It is filamentous bacteria. Understanding the idea or not? Is it clear in the mind? Now if we talk about most of, I am giving information, most of antibiotics obtained from this actinomycetes. Not in NCERT, I am just giving a general information to you. So major source of antibiotic is which category of bacteria? Actinomycetes. Understanding the idea, it is also called filamentous bacteria. Don't confuse with Ascomycetes, that is a fungi. So these all are the example of which particular type of cells? Prokaryotic cell. Understanding the idea? So if we talk about a prokaryotic cell is a smaller than eukaryotic cell. And it multiply rapidly. Why it multiply rapidly? Why it divide rapidly? Anybody can answer? Why eukaryotic cell is slow in division and prokaryotic cell is very fast in division? Any clarity can you give me? Ki prokaryotic cell divides slowly, fastly, eukaryotic cell divides slowly. What is the reason behind it? Because in prokaryotic cell during division what happened? Binary fission. In prokaryotic cell, during division, what happened? Interphase, G1 phase, S phase, G2 phase, a lot of things you were studying, na? Prophase, metaphase, anaphase, telophase, that all mechanism not occur. Simply DNA replicate and divide. Two cell divide. It is binary fission. And that is the reason it is dividing very fastly. And that mechanism of all event of the cell cycle require in which cell? Eukaryotic cell. So relatively their division process is which one? Slower. Got the idea? Now I am going to talk about, beside of different prokaryotic cell, have different shape, their fundamental structure are similar. Beside of different prokaryotic cell have different shape, but who is common in a prokaryotic cell? Their fundamental structure is similar. So if we talk about shape of the prokaryotic cell, what are the possible shape is there? It may be rod shape. When prokaryotic cell have rod shape, it is called bacillus. It may have a spherical shape. When it have a spherical shape, it is called which one? Coccus. It may have comma shape. Comma shape. Comma shape is called which one? Vibrio. It may have a spiral shape. A spiral shape. A spiral shape prokaryotic cell is called a spirillum. A spirillum. Got the idea? So what we are finding? Prokaryotic cell may have different shape, but fundamental structure are similar in which one? Prokaryotic cell. And what are that fundamental structure in the prokaryotic cell? We will discuss in next class. So in next class, what we will finish? We will start and finish the prokaryotic cell, means we will talk about the structure of prokaryotic cell, what are component of prokaryotic cell, everything we will finish. Able to understand or not? And even maybe if time is there, we will enter in which cell? Eukaryotic cell type. Understanding the idea? So bye-bye. Thank you very much for your patience, for listening. Right? Be motivated, remain motivated, and read it. Everything will happen great. Okay, ji, bye-bye, bye-bye, bye-bye. So QR code. So good night. Thank you very much. This is your QR code.